So our last talk is an abstract selected talk. It's another graduate student from this Gerwick Lab. And this talk from the University of California at San Diego is focused on characterizing cyanobacterial hydrocarbons and biosynthetic pathways.
Great, thank you so much for the invitation. It's an honor to be speaking about my work on cyanobacterial hydrocarbons. I'm showing here a marine filamentous cyanobacteria, Morea producens, that we collected in Curacao, and a couple hydrocarbons that are commonly observed in cyanobacteria. And I first want to introduce cyanobacteria to you. Many of you are probably very familiar with cyanos, but they produce about 30% of the Earth's oxygen and contribute about 50% to primary productivity in the oceans, and are highly metabolically and morphologically diverse. And there are a variety of genetic tools available for working with cyanobacteria, and they have a really wide GC content and genome size. In our lab, we focus mostly on marine filamentous cyanobacteria, but there's quite a wide diversity of cyanobacteria that are out there, and they live in all habitats that are on the Earth, and so it's quite a diverse group of organisms to work with. But they're interesting for a biofuel perspective for a variety of different reasons. One, because they're photosynthetic, but also because they produce hydrocarbons. So these hydrocarbons are directly fuel relevant, and the alkanes could be drop-in fuels directly into a diesel or jet engine. Terminal alkenes, however, also have applications for rubber or plastics or even a variety of other lubricants or other fine chemical applications. The branched hydrocarbons also could be more applicable when considering maybe moving more towards a gasoline fuel if you were to break them up into smaller fragments. But the cyanobacteria don't produce quite a lot of hydrocarbons. It's about 0.3% of the overall biomass. So if one were to understand the pathways a little bit more, as well as understand the genes that are involved, you could envision engineering cyanobacteria to produce either more, or taking those pathways and producing them in other model strains. So the physiological and ecological or biological role of hydrocarbons is unknown in cyanobacteria. And so this is one thing that we want to gain a little bit of insight into from our project. And the distribution of the production of hydrocarbons is unknown. And the pathways were only recently discovered. It was really previously, before just a few years ago, we didn't know how cyanobacteria were making hydrocarbons. But they were observed very early on, back in the 60s and 70s. Melvin Calvin here at UC Berkeley was one of the first researchers to look at cyanobacterial hydrocarbon production and even branched chain biosynthesis. So one of the pathways that's involved in producing hydrocarbons is the olefin synthase pathway. That's what the OLS stands for. It's a polyketide synthase type pathway that takes a fatty acid and elongates. The red that you're seeing here is 2 additional carbons that elongate that fatty acid, or fatty acyl ACP is the preferred substrate, to a terminal alkene. Decarboxylation happens in the thioesterase, and you end up with a double bond at the end of the hydrocarbon, the alkene or olefin. And some representative hydrocarbons are shown here that possess that terminal alkene. So that's a signature that one can look for identifying which pathway was producing which particular type of hydrocarbon. The other pathway that cyanobacteria have is the FAR-ADO pathway, and that involves a fatty acyl ACP reductase and aldehyde deformylating oxygenase that can produce a fatty aldehyde and then a saturated alkane. And some representative hydrocarbons are shown here, but they're obviously saturated alkanes, and if there is an unsaturation, it should be an internal double bond, and it comes from an unsaturated fatty acid. So these different structures that are observed are indicative of what particular pathway is observed. But there are two separate, very distinct pathways that don't have common enzymatic mechanisms. And so we were curious about the distribution of these pathways, as well as the composition of the hydrocarbons that are produced from each of these various pathways across a wide variety of cyanobacterial phylogeny. So the project that we set out to do is look at that evolutionary history of the cyanobacterial hydrocarbon pathways, knowing that we have 2 separate pathways to look at, and to characterize the hydrocarbon composition across a wide phylogenetic distribution of cyanobacteria. So this is a 16S tree that you're not supposed to be able to read the titles, but the topology is what matters here. And what I've shown here is the 16S phylogeny of all genome sequenced publicly available cyanobacterial genomes, as well as a group of cyanobacterial genomes that we sequenced from our lab, as well as some from colleagues that weren't publicly available, and a variety of unsequenced strains that represent clades that were not represented in the genome sequenced strains. We have them color-coded by branch according to their morphotype. So we have unicellular and filamentous, as well as heterocyst-producing and the branching strains, and color-coded the names of the cyanobacteria according to which pathway they possess. So the blue being the FAR ADO pathway, the red are the OLS pathway, the olefin synthase pathway, and purple being those that we don't have a genome for, and so we don't know which pathway they possess. So we were interested in looking at the distribution of these two pathways. So we queried each genome for their biosynthetic pathways, and we found 119 homologs of the FAR ADO pathway and 17 homologs of the OLS pathway. So going back to the tree, we can see that the most widely distributed pathway is the FAR ADO pathway, and likely the ancestral pathway for hydrocarbon biosynthesis in cyanobacteria. And the more narrow distributed pathway is the OLS pathway, the red pathway that's shown here. And we notice very quickly these strains that are not very closely related to this main clade in here that possess the OLS pathway. And to zoom in on this just a little bit more so that you can hopefully distinguish which strains we're looking at, Morea is the strain that I showed in my first slide, is up here. And this is the marine filamentous strain. And Leptolingbia PCC 6406 is down here, very distantly related. So we were interested in finding out whether horizontal gene transfer was involved in the evolutionary history of these pathways. So when considering the criteria that one would look at for considering horizontal gene transfer, you would want to consider the definitions according to the movement of genetic material other than my traditional transfer or vertical transmission of those genes. So that would involve looking at the phylogenetic tree or the topology of the genomes, patchy pattern of gene loss, and the GC content or the presence of transposons, as well as the disruption of the synteny in the genome. So I'm going to go through a couple lines of evidence, but I don't have time to go through all of them. And first, I'll show you the GC content information. So the genome GC content of all the strains that have the OLS pathway are shown on the left, and the OLS gene GC content is shown on the right. So a pattern emerges that the OLS GC content is just slightly higher than the genome GC content for all strains. But we also see that the Leptolingbia strain has quite a high GC content for its OLS pathway at 64%. And looking at the gene neighborhood using IMG, another pattern emerged that was unique. This is the gene neighborhood for the FAR ADO pathway in Senecacoccus 7942. And we see these 5 genes found in almost 70% of genomes of cyanobacteria. And when we consider the gene neighborhood for a couple strains that possess the OLS pathway, we actually observe that strains in that same gene neighborhood in very close proximity. So this acyl-CoA carboxylase that's just downstream is also found in very close proximity to 4 of the strains that possess the OLS pathway. We also see the short-chain dehydrogenase and the GTP hydrolase in some. We also see the presence of transposases and phage assembly proteins here. So when considering the different criteria for horizontal gene transfer, I didn't have time to show you the topology of the genes that are found, but we see the Morea and Leptolingbia OLS pathways clading together as opposed to distantly as they are in the 16S tree. And the patchy pattern of gene loss is also observed, and presence of transposons and different GC content in the genome and disruption of synteny certainly are matching our criteria for considering the role of horizontal gene transfer for this pathway. So we certainly can say that horizontal gene transfer likely occurred at this stage, but the involvement of horizontal gene transfer in this clade is unclear. However, the disruption of synteny that's in the remaining 30% of cyanobacterial genomes is mostly found within this clade. And so I think that there's a really deep evolutionary investigation that could be done to really query the evolutionary history of the OLS pathway within this clade as well as this clade up here. And not very much sequencing effort has been done with some of those those strains. So there's quite a lot more that we can learn about the evolutionary history of this pathway. So I also was interested in looking at the composition of hydrocarbons found across the wide phylogeny of cyanobacteria. And the main question was, how widespread is hydrocarbon production? Before this study, we didn't know whether all cyanobacteria produce hydrocarbons. And we also were interested to see if certain cyanobacteria produce different compositions. And it wasn't clear at that stage whether the few strains that had been queried were the diversity had been completely characterized from hydrocarbon composition. And we were also interested in the prevalence of modified hydrocarbons, so double bonds and methyl groups, and whether those can tell us something about the biosynthetic pathways that are involved. So going back to our 16S phylogenetic tree, we selected strains across the wide phylogenetic distribution of cyanobacteria. We worked with 32 strains of cyanobacteria to grow them up and characterize their hydrocarbon composition. In using conditions that are appropriate for each strain, we dried the biomass and did the extraction and characterization with GCMS analysis and structure determination using authentic standards when available and derivatization to determine the double bond position for these hydrocarbons. We also use an internal standard for quantification of the overall biomass percentage. So I'm going to delve right into this. There's a lot of information here, and I'd be happy to get more into it with anybody that's interested. But these are all the strains that we looked at. And there's their phylogenetic relationship on the far left and the percentage of total hydrocarbons for each strain and the type of hydrocarbon that's found. We have the different grouping of different hydrocarbons over on the far right. But one can see very clearly that blue is the most prevalent hydrocarbon, and that's heptadecane. And so that's a 17-carbon hydrocarbon that comes from the FAR-ADO pathway. And that's to be expected because that pathway is very prominent. And 18-carbon fatty acids is the most common fatty acid that's found in cyanobacteria. We also observe, however, some branched hydrocarbons. So these strains are all in the Nosticales. These are heterocyst-producing cyanobacteria, and they all seem to be producing branched hydrocarbons, and that's unique to this strain with the exception of Gloeobacter, which is a fairly primitive cyanobacteria, and this is the first observation of it producing branched hydrocarbons. So we are very interested in looking at the methyltransferase that we would predict would be involved in the biosynthesis of branched hydrocarbons in cyanobacteria. That brown and yellow up here is also branched hydrocarbons. We observe this clear different composition from strains that possess the OLS pathway, and those strains obviously produce terminal alkenes, so that red is the 1-heptadecene, but the purple and gray and yellow that we're observing there, these are internal double bonds. These are 2, 3, and 4-heptadecene, and these are inconsistent with our known mechanism of the OLS pathway that would produce a terminal double bond. So that isomerase or different desaturase is what we would hypothesize would be involved there. So there's a variety of questions that come about because of this characterization of the hydrocarbon composition from cyanobacteria that's new to our understanding of hydrocarbon biosynthesis in cyanobacteria. So I did the quantitative analysis, and we see the range of overall hydrocarbon production within range of 0.02 up to 0.26. And that's within the range that was previously reported of about 0.025 up to 0.1. It's a little over the previously known amount, but we also see those with the OLS pathway possessing more hydrocarbons as a total percent dry biomass than those with the FAR-ADO pathway. And that's statistically significant. Those with the OLS pathway produce more. But the caveat is that that does not account for growth rate. And some of those strains do grow rather slowly. So we still have yet some more to do to characterize the overall productivity between these strains. So in summary, we see that hydrocarbon production is universal among cyanobacteria, and this is not known before this study. And it's a unique observation that something so fundamental to cyanobacteria is not very well understood. And we think that the production of hydrocarbons for cyanobacteria has had some major selective pressure to maintain one or the other pathway, and we don't know what that selective pressure is. We might hypothesize that cyanobacteria are all photosynthetic, and it could be quite likely that, say, the thylakoid membrane possesses those hydrocarbons, and we can easily do that test. And we might hypothesize that it could be another chemical signaling molecule. It could prevent desiccation or be an energy density storage molecule. All these are testable hypotheses, but we still don't know. And this is a very interesting observation and could inform someone that would be interested in not only understanding cyanobacterial physiology more, but also using these pathways for biosynthesis biofuel perspective and understanding how you can modify those strains in the future or other strains that would be producing these types of hydrocarbons. The OLS pathway evolved involving horizontal gene transfer, and the OLS pathway strains seem to produce a higher percentage dry biomass than those with the FAR ADO pathway. And terminal alkenes are found only with strains with the OLS pathway. That's something that we predicted and we observed as we went in to do the characterization. And branched hydrocarbons are produced in only a small subset of cyanobacteria. And we're actively involved in identifying the methyltransferase that we think is involved in that pathway with Anabina 7120, a strain that has quite a lot of genetic tools available to do that work. So with knockouts and expression, we hope to be able to characterize branched hydrocarbon biosynthesis. And presumably you could apply that towards gasoline production with branched hydrocarbons being more relevant for that application. So with that, I'd like to thank my colleagues in the Gerwick Lab at Scripps Institution of Oceanography at UCSD, as well as my co-authors. Dr. Ala Lapidus is here at the conference, my thesis committee, and my colleagues on this work and the funding. And with that, I'd like to thank you for your attention. That's the publication. Sorry, I forgot to mention.
Are there any questions? Okay.